Recording in progress.
Once again, good morning. Today we are here to discuss about the plant disease management. Even though we have already, all of us, we have already observed, maybe we had an experience with dealing with plant diseases. It is better to start with some kind of a preliminary introduction to plant diseases, which is known to us, but the details of how they are developed, why should we consider it as issue or problem and something to be managed, and if it is necessary to be managed, what are the methods to be applied to control the diseases. So, as we can see in the picture, we have seen these types of problematic appearances of plant organs, and sometimes even without considering what is that, how it has been developed, we could have maybe taken some actions to control these problems. Now, maybe we could achieve success of managing the problems, but knowing the background of these diseases or these problems, then we can do the management or control more efficiently with maybe a lower cost and sustainable management of the problem. So, when we look at these plant diseases or the organs, parts of the plants with the diseases, we can understand that will definitely create some damages, and ultimately there can be a loss of the outcome or the product or the yield. So the average yield loss due to the plant diseases is around 10 to 15%. That is under the normal spread of a disease. But if the disease is at severe level or at higher level, there can be maybe losses up to 20 to 30%. Then that is during the lifespan of the plant or before harvest. Then during the post-harvest handling, after harvesting, there can be additional losses due to post-harvest diseases. Now, these are the amounts in the world, average amounts in the world, but when it comes to our country, the losses are, of course, higher than the average yield losses due to diseases in the world. Now in some reports says that especially in vegetable and fruit sector in our country, the yield losses during the cultivation is around 30, And if it is exposed to poor handling after harvest, then the another additional 20%, that means around 50 to 60% of the yield is lost due to the plant diseases. So it is a severe problem. Now we can see that then there is a necessity of controlling plant diseases or managing plant diseases. So we can see here in this picture, in these plants, that means in rice plants of this picture, on the leaves there are some disease or diseases are spreading on these plants. And then here also on these fruits there are problems that can be considered as diseases. Now here the leaves are showing some symptoms of the diseases, and here the fruits are showing some symptoms. So we can go into the details of these problems later on. Now just for the time being, these paddy fields with this type of a problem will be having a reduced yield than a normal healthy paddy field. We can accept that. And the fruits that are having these types of problems will not be accepted to the market. Even at home, maybe from our garden, we will not be consuming these types of fruits because of the available diseases. As these losses will be reducing our income, reducing the profit, we will be taking actions to control the diseases. So for the control of the diseases, we are spending huge amounts of money in the world in order to control fungal diseases. We are using around, or we are spending around $7 billion for controlling the diseases caused by fungi. Annually, we spend this huge amount. So this is some average number. It can be higher than this or a little bit lower than this. Anyway, this is a serious amount. In addition to the money we are spending to control the diseases, we have some issues of social, cultural problems created by plant diseases. Now, when there are symptoms like this appearing on the plant leaves, crop leaves, the farmers are, of course, thinking about the necessity of controlling the diseases. So their normal cultural practices will be changed, or they have to change some cultural practices to control the diseases. Then in addition to that, if there are diseases like this, harvest will be reduced and the income of the farmer will be reduced, or else the food supply to the farmer and to the society will be reduced. Then that will be having an impact on our cultural aspects like festivals. Now we have the post-harvest rituals, festivals, offerings to various religious institutions or offerings to other people. So we can't do those things as we would like to do if there are no yield or outcome due to the plant diseases. Therefore, plant diseases are having serious negative impact on our life. Expenses and the changes impact on our life. Then when the diseases are at higher level, it may cause the tragic or disastrous situations in our society, in our region, in our country, or in the world. So, when the diseases are destroying the yield, no food will be available to people, then that may be a cause of death of higher number of people. So these, some of the incidents, examples of such disasters caused by plant diseases are recorded in the history. They are not much pleasant to hear, but just to make us thinking about seriously, thinking about the plant disease management, I would like to give some examples of such disasters caused by plant diseases. So, one of the frequently mentioned problem created by the plant diseases is the so-called potato famine in Ireland, 1845. The potato fields of Ireland were infected with potato late blight disease, and due to that disease, potato yield was destroyed and people were suffering from hunger. They have not received any food because the potato is their staple food like rice for us. So millions of people died. You can read about that if you are searching information on Irish potato famine. Then another frequently mentioned example of disasters, drastic changes in the society or dramatic events in the life of people, changing economy of a country was happened due to an epidemic condition of a plant disease. What is this plant? The coffee plant. Coffee plant is having many diseases. One of those is the coffee rust disease, which is appearing like this. So the leaves are destroyed and this yellow color, cottony-like appearance developing on the lower surface of the leaves normally, and that will destroy the plant, destroy the yield and the income of the planter or grower of coffee. So, as you know that British people, when they got the authority of our country, they have started some changes of agriculture and they established coffee plantations instead of other crops we have produced at that time. Then when the coffee plantations were affected by this disease, the planters, they couldn't control it. And they started thinking about some alternative income sources for their plantations. So ultimately, they started replacement of coffee in their plantations with tea. So that is how Sri Lanka, which was a coffee producing country in Asia or in the world under the British Empire, we gradually changed our country into the country of producing tea. So it's good that we found another alternate crop that is suitable for cultivation in our country. But if there was no epidemic of coffee rust disease in Ceylon at that time, we we are considered as a coffee producing country up to now as well. Sri Lankan Ceylonese coffee was famous in Europe at that time. So maybe you wonder why these pictures of two books, the title pages of two books, and a photograph of a lady is appearing on the screen. Now, these two novels, these are not scientific publications. These are two translations of the same novel written by this author, Christine Wilson, this lady. If you are interested in literature, maybe you have heard about a British writer who lived in Sri Lanka, who was an officer, medical officer, Dr. R.L. Spetley, and he had written many famous novels, and this is his daughter. While the father was traveling in Sri Lanka in search of helping, in search of finding needy people to help. At the same time, he found the clues for starting his novels or literature creations by Dr. R.L. Spetley. So his daughter was observing what father was doing, and she also started writing at her age. So why I'm putting these two novels are actually one novel. I'm just referring to the Bitterberry novel, famous novel written by this lady, is discussing about the change of social behavior, life of people at that time, and the background of those stories were the period of spreading coffee rust disease and replacement of coffee with tea at the end of the story. So, no harm. Actually, it will be a benefit if you read finding these books from a library or by purchasing. It is an older version now. So there were many translations other than these two. I am talking about a book because of two reasons. One is it is good to recommend you to read this type of literature as a relief from your schedule of studies, so to give you some relaxation. If you are really involved in studies with a lot of enthusiasm, then you may need to have a break. And whatever the reading will be beneficial, and this one will be more beneficial because it is explaining the development of coffee rust disease in connection with the behavior of planters, their families, like that. Okay. So that was about an epidemic of plant disease in Ceylon. Then there were examples of such in other countries, and here an example from our neighboring country, India. In Bengal, they had to face a famine in 1940s due to the spread of the cereal crop disease, brown spot disease. So again, millions of people were dying. So now these are some evidences to say that plant disease management is essential. We need to carry out the disease management carefully and efficiently, otherwise there can be a catastrophe. So now we can accept that there is a possibility of threat for our lives because of a plant disease. And in addition to that, there can be a threat of changing the economy of the country due to the spread of plant diseases. So going back to the diseases and their losses, I would like to request from you to help me to define a disease or explain how the yield is reduced. Now, these two photographs are, of course, explaining us how the disease is reducing our yield. So for me, when I look at these fruits produced by these tomato plants, when they are infected like this, I can understand that those tomato fruits with these types of diseases will not be accepted by consumer. So I will be losing my market income from the selling of these tomatoes. I know that the disease is destroying the tomato fruits here. So my harvest is tomato fruits. And if I lose tomato fruits due to the disease like this, maybe I would like to harvest these tomatoes and send to market, but I have to remove, or at least the buyer will be removing the badly damaged fruits. So the disease is reducing the production of fruits, which is the important part of my harvest. I have a paddy field and I can see some diseases are spreading on the leaves. As the disease is spreading on the leaves, my feeling is that I should not worry about the spread of this disease, because in my tomato field, I can see that tomato fruits are infected and my yield will be destroyed by this disease. But in my paddy field, the disease is there, spreading on the leaves. The panicle is developed. The panicle is, assuming that the panicles of these rice plants are not showing any kind of a disease or disease symptom. So maybe I can ignore this because the leaves are not harvested by me. I am harvesting the grains, rice grains. Therefore, I am not worrying about the spread of this disease. I think you can see the changes of the colors on these leaves. Now, if I am doing something wrong. Maybe you can help me to correct myself. So, am I correct when I'm thinking that this is not causing any threat to my rice yield? And here I am thinking that these fruits will be causing a reduced income from tomato field. But I expect the usual income from my paddy field. So take 2-3 minutes and tell me whether I am correct or not. And also let me know why you say I am correct or not correct. Any volunteers, please? To help me to take a decision on the necessity of controlling this situation in this rice field. Yes. So can I request some of you? Teshan, can you please? Is the question clear? So Teshan is not here, maybe. Then Chamod. Nasreen, what about you? So you are not willing to help me?
Excuse me, sir, if the disease spreads during the harvesting, so that will retard the growth of the plant. So as a result, it will reduce the yield.
Now, what you say is now, if the disease is at the harvest, reduce the growth of the plant, or
During the growing period,
during the growing period, if there is a disease,
that will deaccelerate the growth of the plant.
Okay.
And as a result, it will reduce the yield.
Right. Okay. So if the plant growth is affected by this disease, for an example, then it will be reducing the yield. Now, if I just argue with you that now this plant is now it has reached the maybe maximum level of vegetative growth. It started flowering and now panicle is developed. So there is no further growth of the plant is required. Now the growth of the grain is required, and this disease is having effect on the leaves, not on the panicle or the grain. So how would you argue with me in that case? So no growth is expected now from this plant, only the panicle growth, and the panicle is not affected by the disease.
Then there won't be any reduction of the yield.
Because there is no damage by the fungus to this panicle. Right, that is possible. Any other opinion? Now, there will not be any direct damage to these panicles by the disease. But there is another issue. Now, the disease is spreading on the leaves, and the leaves are not harvested by the farmer. Farmer is expecting the good quality grains, and the grain is filled with stored food from where it get food to store in the grain. I could ask the name number 3027. You were starting logically, very good point was there. Can you please come back again?
Yes, sir.
Yes. Your name, please, first.
My name is Hudson.
Okay, Mr. Hudson. So, Hudson, what do you think about now that point? Now, the grains are developing now, and it has to be filled with stored food. From which part of the plant, or how the food is filled in this grain? It is filled with the carbohydrates, starch or sugar that is produced on the leaves. Leaves are doing photosynthesis, and the extra photosynthate produced in the plant. Why it is extra? Now plant is using the food produced by the leaves for respiration and other functions require energy. Energy is given to the plant by the product developed during the photosynthesis. So, the apparatus of photosynthesis is the plant leaves. If the leaves are having a disease, even though it is not harvested by us, so the extra food produced by the leaves will be translocated into these grains, right? So, grain filling will be reduced by the fungus or by this disease. Indirectly, it will be reducing the yield. Agree with me, Hudson?
Yes, sir.
Right. So, now these two photographs gives us that idea. Now, some diseases are causing damages directly to the harvested part, and we name those damages as direct damage. Later on, we will talk about this again maybe. But for the time being, I think better if you note down, the diseases are causing two types of damages. One is direct damage. That means even a pest, an insect pest, is also doing the same sometimes. So the damage is done to the harvested part, and that is direct damage by the pest or the disease to the yield, direct damage to the yield. But here, it is not direct damage, it is an indirect damage to the yield, but yield loss will be maybe serious like this as well. Why? The grains will not be filled with stored food if the stored food producing apparatus leaves are affected by the disease. So, that type of damage is considered as indirect damage to the yield, but that is also serious, right? So, we can go ahead now. Now we know what is the damage or what are the damages done by these diseases to the plants. So the plant diseases that are causing these types of disasters are caused by different groups of organisms. One type, fungus or fungi, that is one group. Now, they are microorganisms that are causing plant diseases, fungal plant diseases. So, we can see that some fungal parts are developed on our plant product, on the stem of a plant or on fruits of plant. So if you observe the plant tissues infected by the pathogen, we can see these types of structures of the fungus inside the host or on the surface of the host. Then there are some primitive organisms like the protist. They are from the class Protista, another group of microorganisms. But the most commonly causing organism group is fungi. For a certain extent, smaller number of diseases, and even though we don't have examples of such common diseases caused by protist, but there are diseases caused by bacteria. So there are bacterial leaf or other parts diseases. You can remember a plant pathogenic bacteria found in that organic fertilizer ship imported from another country. Our authorities, they have tested the organic fertilizer in that ship, and they found in the samples tested, there were evidence of presence of a bacteria called Erwinia. So that is a plant disease causing bacteria, Erwinia, and one type is Erwinia ceratophorum. So anyway, bacteria are causing diseases as well. Then there are other groups of microorganisms, molecules. They are also causing plant diseases. So the difference between molecules and bacteria are that bacteria are having well-formed cell wall around the cell or bacterial cell, but in molecules, the only a membrane covering the inner material is available. No formally developed wall of bacterial, like in bacterial cells. So, when the organisms are without a cell wall, we name them as prokaryotes. Prokaryotic plant pathogens are there. Molecules are having three different types called mycoplasma, phytoplasma, and spiroplasma. Another common group, now most common group is fungi, then there are bacteria, then again another common group, more commonly causing diseases than protists and molecules, the viruses. So viral particles are also creating diseases in plants. They are also very small. You can't even see through the normal optical microscope. You need to use an electron microscope to observe viruses. And it contains a protein capsule and inner material RNA molecule. A relatively larger group of organisms, nematodes, but they are also microscopic. They are also causing diseases. This is the plant pathogenic nematode piercing the plant root hairs, injecting this apparatus the nematodes apparatus of piercing and sucking cells from the plant is named as stylet. The stylet is making us easy to differentiate plant pathogenic nematodes and neutral nematodes who are not causing any damage to the plants. Right. So those were microorganisms, primitive, causing plant diseases, but there are organisms from the higher taxonomic level of plants. Now, maybe you heard about these plants in your school education. And now these are parasites, parasitic plants on other plants. See these thread-like structures, white or brown color. It is a plant. And this plant is having flowers, producing seeds, but we can't see any kind of a leaf because it is a parasitic plant. The plants with leaves, they are not parasites. They produce their own food by their own leaves. But parasitic plants like Cuscuta and some other plants, they absorb nutrients, water from the host plant like this. And when they get mature, they produce flowers, seeds, and the seeds will be disseminated into other plants and starting the diseases. Now, how do you think, why when this type of a plant is grown on our plant, the green color plant we call a host plant, hosting the pathogen. Why this plant is facing a problem? Now this one is these thread-like structures and flowers you can see. Now it is growing, and what will be the result? If it is absorbing water and nutrients from this plant, not only water and nutrients, even the photosynthetic surplus of this plant is absorbed by this Cuscuta or dodder plant. Continuous. Yes, please. I saw that you started asking for something or explaining why the presence of Cuscuta is causing problem to the other plant, this plant. Who was trying to tell me something? Hudson?
Yeah, this parasitic seed plants will absorb the nutrients and water from the host plant. Then it will affect the growth of the host plants, because part of the nutrients and water will be absorbed by the host plant. No, not host plant.
Parasitic plant.
Parasitic plant.
Yeah.
So they will have an impact to the host plant.
Yes. So now one impact is that if it is absorbing water from the xylem of host plant, then no water, sufficient amount of water to the host plant. If it is absorbing food produced from the leaves and translocated into other places through phloem or other parts, then there will be no food, photosynthetic material to the plant because the surplus amounts will be absorbed by the parasitic plant. And on the other hand, this presence of this parasitic plant will be covering the leaves of the host plant. That is also a problem. And of course, evolutionarily, it has become very thin, no leaves, no much coverage, but it is absorbing everything from the host plant, and gradually it means the host plant will be damaged, showing lower yield if it is a crop plant. So this example is from our country, Cuscuta. There is a Sinhalese name. Anybody who can remember, who had observed this?
Agamula නැති වරස?
Yes, Anugamula නැති වැල්. Because he can't see any root system of this plant attached or penetrating in the soil. Then we can't see any terminal end of the plant because it's spread everywhere, all over the canopy of the host plant. So because of that, maybe it has given the name Agamula නැති වැල්. So if we just see the um, groups again: fungi, nematodes, bacteria, molecules, viruses, and parasitic seed plants or plants from higher taxonomic categories. So these different types of pathogens are causing diseases to the plants. Then when they are causing diseases, we can see there are some changes of the plant appearance, many changes. So we have seen many different shapes, problematic appearances of the plants like this. See, this bark is having an unusual color patch like a scar on our skin, cracks on the periderm, or cracks on the stem or appeared plant part, and droopy leaves. Here we can say that it is droopiness, then wilting symptom, dehydration of the leaves. And if we assume that this plant is a healthy, normal plant, see, it is bearing, there are young fruits, ripened fruits, leaves are showing the normal appearance. And if we assume this as a normal, healthy plant, and these are plants of the same species tomato, and it is having a problem, something we can understand. We can understand at least that it is a problem. There is a disease. So this one is also from different species, but it is also showing us the symptom of droopiness. Here there may be many symptoms. Lack of a bark in one place. Right, okay. So here also there is a difference. Now, the question is, what is a plant disease? So, compare this plant with these abnormal plants or plant with a disease. Compare it with this healthy plant, which is having everything normal. And here, it is different. Now, again, I am trying to get your help. Now it is more easier because we have discussed about what is a plant disease earlier. So, can you write down a definition? What is a plant disease? Considering these comparisons. So, have a look at this. Take 1 minute to look at this. Think, at least you can compare easily this healthy plant or plants in some area. So, in the same area, there is another place where plants are grown, but something is wrong. So, have a definition or explain or write down your answer. What is a plant disease? I'll be here to get it from you. Maybe take 2 minutes to think and write the correct definition of a plant disease. I'll invite you to that.
Recording stopped.
Excuse me, sir. My definition is
Okay.
plant disease cause direct or indirect damage to the plant. These
Right.
plant diseases will be caused by microorganisms such as fungus, bacteria, virus, molecules, and parasitic seeds.
Right. Okay. So it's caused damages to the plant and it is caused by the different organisms, microorganisms mentioned by you. Right. Okay. Good start for the definition. So anything to be added to that definition to explain further what is a disease compared to a normal healthy plant? So Rushan Muhammad. Then Krishna. Seems that you are not, either you are not here, just you are logged into the session, but away from the computer or phone or device, not in touch.
Excuse me, sir.
Please.
Sir, plant disease is systematic, abnormal, psychological function of a plant.
Abnormal physiological functions of the plant.
Sorry, physiological.
Yeah. Yes. So yes. So that is the thing we can add into the previous definition given by your friend. So Anushka is telling that because of a plant disease, there can be, or during the plant disease, there is an abnormal functioning of the plants, specifically the physiological functions. So the normal physiology is not there. There is a deviation from the normal functions of the plant. So, yes, now it is perfect, the definition. So, now considering these two tomato plants, now what are the abnormal functions of these plants compared to this normal plant? It may not be reasonable to ask from the same person again and again, but as you are the, creator, maybe you can elaborate, Anushka. Now, what are the abnormal functions of these plants compared to these normal plants? When you look at this, I think picture is clear.
Sir, regarding this normal tomato plant, their leaf is very high amount of the growing rate. But when we consider the another another tomato plant, their leaf is very low amount of the growing rate, sir.
So it is low grown or having abnormal appearance. These leaves are droopy. And because of that, now the sunlight interception by this plant is reduced. And I would like to get that. You said some physiological functions are abnormal. Can you name some physiological functions? You said abnormal growth, poorly grown. That is true. Now the functions of these leaves of this plant, what are the functions affected by the disease?
I think so photosynthesis is affected.
Photosynthesis is affected because the sunlight is not intercepted properly, because leaf blade is droopy downward, and it may not be receiving sunlight well. Photosynthesis is affected. So for normal photosynthesis is not there. That function is deviated from normal condition. Right. Then this is actually a plant affected by the wilt condition. That means lack of water. It may be something happened and the water supply is not there. So the transpiration is poor. It is affected. Again, the transpiration is deviated from the normal functioning. So if the transpiration is deviated, if it is abnormal, now we have already mentioned two: photosynthesis, transpiration is affected. And that will be also creating another deviation. What is transpiration? Absorption of water, and specifically the movement of water through the plant and release of water to the atmosphere through stomates. Transpiration. It is a kind of water release from the plant, but it is not just releasing water or something like removing water. That water flow is maintained as a circular, just a halfway circulation of material. So nutrients absorbed from the roots are taken to the aerial parts because of the transpiration. Because of the transpiration, the water is given to the leaves, and not only water, elements, nutrients are transported to leaf or other aerial parts by transpiration flow. So that is affected now. If transpiration is not there, no flow of nutrients and water. And if the nutrients are not there, again, it will be creating problem for photosynthesis. And transpiration flow make plant steady. It will keep the turgo pressure. Water is given to the cells and tissues by transpiration. So that is another deviation from the normal functioning. Then if the photosynthesis is now reduced, then there will be lower quantity of energy source will be produced by the plant. If the energy sources are reduced, then the respiration will be affected by the diseased plant or affected plants by these diseases. So now we have not mentioned maybe all and every deviations. Now I am giving you the task. It is up to you to think what are the physiological, biological functions of the plants like this, deviated from normal functioning. So abnormal functions are there. I think if we are to discuss the abnormal functions of the diseased or infected plants, it may take the maybe full day to talk about that. So because of that, we temporarily suspend that discussion, accepting that there are many biological, physiological functions of the plants are deviated from normal functioning, right? So now there is another thing we have to consider when we are talking about plant diseases. Now, when you are looking at these plants, now not the plant in the center, this plant with this scar on the stem bar, this plant with some cracks on this bark part, these plants with some droopy leaves, these plants are also with some droopy leaves, and this is a normal plant. Now, we all considered these, these four surrounding plants are as a plant or plants with some disease symptoms. Why is that? So I'll give my answer. I think when I'm looking at these plants, they are different from this normal plant. They are abnormal. These plants are showing something unusual to me. And because of that, I consider they as abnormal plants. And when I'm telling like that, when the plants are showing these type of droopy leaves, I think about 2 months ago, in almost every part of our country, during the daytime, many crop plants were showing this type of appearance during the day because we couldn't provide water as we wanted. So we saw these types of changes appearing on our plants. But in the evening, these leaves become normal like this. Then that type of temporary change of the plant appearance, will it consider as a disease? So I am deciding that these plants are infected or having a disease because of this change. Then, yes, anything you would like to mention? So, as you are silent, so what to do? I have to continue. So,
I think, sir, there are some trees, this will become like this tomato trees. In the evening, they will become okay. Then that will not be a disease, I think.
yes.
That will not be a disease.
Right. So you don't consider it as a disease. So it is a deviation from the normal conditions temporarily. So
yes, sir. Yes.
Okay. So when the problem is solved in the evening, maybe the high temperature, lower water supply, due to higher temperature, plant is trying to reduce the water loss by making the leaves droopy, closing the stomates. It is showing the reduced transpiration. It make lower turbo pressure, and plants are showing these wilting symptoms. But if the appearance is coming back to normal during a short period of time, then we don't consider it as a disease condition. So, deviation from normal functioning for a particular longer or relatively longer period of time can be considered as a plant disease. And to indicate that there is a disease, sometimes plants are showing or expressing some signals are given. They indicate that they are sick. So when we are infected by viruses or bacteria, or sometimes whenever we have a disease in our body, maybe we don't see what is the disease. We don't see viruses in our lungs, inside our body. But we can feel that our body temperature is higher. So we say that, I am having temperature. Maybe there is a disease. So high temperature is not the disease I am having. It is an indicator of the disease. It is a signal given by my body to say that you have a disease. And those visual expression of the plant diseases are considered as symptoms of plant diseases. So, if we see that, so the expressions, visual expression of the presence of a disease is considered as symptom of plant disease. See these lesions or leaf spots, leaf patches, lesions. Reducing the green color is named as chlorosis. The lesions, necrosis means the death of the cells. We can see the dead cells or tissues due to diseases, so caused by the blight disease. Serious blight. Extensive, we call the now, when few leaves are showing symptoms of blight, it looks like this. Some parts of the plant leaves are becoming light brown, dark brown patches in this area, light blight disease. Then when those patches are amalgamated, forming maybe blighted leaves, we called it as extensive blight. Sudden necrosis means like this. Sudden death. Necrosis means death of something. Necrosis of leaves, necrosis of stem. necrosis of fruits like that. Another type of changes, chlorosis, reduced amounts of green color is there. Not only yellowing, there can be anthocyanin pigment color. So due to we mentioned that there is a lesion called chlorosis lesions. When the green color is reduced, sometimes the production of anthocyanin, which gives the reddish color appearance, is there. So it is now instead of green, red is appearing. That means tannin is anthocyanin is developed, or other material will be developed because of the lack of chlorophyll. Then chlorophyll production will be also affected if there is a pest or pathogen is suppressing the production of chlorophyll. So on the leaf blade, there will be areas of dark and light green color patches, or sometimes yellowing and yellow and other color pigments are appearing alternatively in different places of the plant leaf. So that is mosaic symptom. Change of shape like this called rugosity. Then some changes or excessive growth of some tissues and organs are there. We name the production of such excessive production is named as hypertrophy. And as a result of hypertrophy, the galls are produced. We call these types of unorganized cell mass developed in attacked places called gall formation. So these types of symptoms are appearing on the plants. So by looking at the symptoms, we can identify the diseases as well. So the nodules are developed. Sometimes the beneficial nodules are developed by rhizobium to fix the nitrogen. But some pathogens are also creating a nodule type developments, and they are, so the real root nodules of legume plants in symbiotic relationship will be beneficial, but the development of nodules by other microorganisms other than the rhizobium, there can be problematic, and they are named as rhizobium is good, but the galls produced by other bacteria like Pseudomonas tumefaciens is a serious problem. Then another common type, lesions on the seedlings, they are like this. So the lesions of seedlings on the collar region like this, we name the disease as damping off. Then the lesions on mature plant parts like this, we call them as concave lesions. The cankers on the stem or plant parts. Then the not only these stems or roots are affected or leaves are affected, sometimes the reproductive organs like fruits, floriculture parts, so we call inflorescence, flowers, yams, tubers, flowers are with some specific symptoms like this. Another specific type of symptom, disruption of reproduction, floral parts are affected and they are replaced by vegetative structures. Floral parts are replaced. So instead of having flowers, the plant is producing stems or shoots. We called it as phyllody condition. Sometimes the overgrowth of plants, maybe we will think like that, but the reality is that some plants are with lower growth rate. We call hypertrophic symptoms. And if you can remember earlier, we saw that the dehydration symptoms, the wilt, and that means the dehydration, droopiness, it may caused by many pathogens like nematodes damaging roots, fungi or bacteria are blocking the xylem tissues, and maybe lack of water in the medium or soil. So those are some reasons for the symptom of wilt in the plants, right? So during this part up to now, we were talking about what are the plant diseases. We have defined it. Then the general discussion on how plants are developing symptoms. That was our part when we were going through the definition of the plant diseases, right? So now we will have a break for 15 minutes. I will continue with the next part of today's session. So I can see that some of you have started leaving, but I'm going to continue. My recommendation is to be connected. Otherwise, today, I don't know how about in your area, due to the poor weather conditions, there will be a lower rate of connectivity to our session. So we will meet again after 15 minutes. So have a break, and after that we will continue.
Recording stopped.
Due to the sudden visit of few my research students from the external degree program, so they are here for an examination. Anyway, in between the two exams they came, so I thought of just give some brief instruction to them. So anyway, the number also in your now before the break we had around 75. Now it has been dropped to 64 down. Anyway, I think we will continue. Next thing we need to understand is that now we know about a plant disease. What is a plant disease? how it is affecting the production, and we know why it is necessary to control the diseases. Otherwise, there will be a yield reduction. So because of that, we need to manage the plant diseases. So for the management, it is essential for us to identify the plant diseases, understand, identify what is the disease, what is the causal organism, what is the reason for the disease, and because of that, now, if we know the reason of the disease, then only we can do the correct management. So, because of that, we have selected this as our next topic.
Recording in progress.
The second part of the today's session is on classification and identification of plant diseases. So, classifying something, grouping the problem according to the characteristics of the problem will be giving us a higher potential, better opportunities to control that problem more efficiently. Same with the plant diseases also. So, when we consider the diseases, there are two types of diseases in the plants. It is similar to the diseases of animals, animals like mammalians, so animals like human being, we are also the living being. So, there are two types of diseases we have and the plants. So, the first type is non-infective diseases. That means the diseases are not transmitted from the infected or diseased organism and to other organisms. So, now it is something like that. Now we have communicable diseases and non-communicable diseases. We say like that. Now, in our life, we are frequently having the problem of viral infections, influenza virus, or some other those types of diseases. When maybe somebody in our circle of activity, when one of your friend who is closely associated with you, maybe a friend in your batch, when they are coming to the sessions in lecture halls or in laboratory, there is a possibility of transmission of an infected disease and influenza virus from one person to another person. Last month, there was an epidemic of viral influenza in our university, and we had to close the hostels for 1 week to prevent the spread of that disease among the students because it was transmitted. Then we know that there is a common disease problem among our people. Now there are different types of heart diseases, heart problems. So I'm not going to talk about what are the heart problems, but we know that a heart problem of one person is not transmitted, not communicated to another person from that person with heart disease. So we call that as non-communicable disease. So I told about that to give you the idea, you all know about it, that the plants are also having non-infective diseases. Those diseases are not caused by pathogens like bacteria, virus, fungi. So it is not transmitted. Mainly those diseases are caused by the abiotic factors. That means non-living factors like lack of nutrients or nutrient deficiency. You know that the lack of nitrogen may cause yellowing, chlorosis of the leaves. Like that, it may be a problem, disease, because that deviation from normal functioning of chlorophyll means photosynthesis. It is not temporarily. If the nitrogen is not there, there will be malfunctioning of photosynthesis. So that is due to nutrient deficiency. Then if the nutrient is at excessive level, it may be creating nutrient toxicity. So you were talking about plant nutrients earlier. I am not going to talk about it again and again. Then same with the water relations. Sometimes when water is lacking, the plant is showing wilting symptoms. Drought condition, lack of water will create wilting of the plants. So excessive water may create also some problems of the plants. So those types of diseases are non-infective. They are also to be managed, but we are not going to discuss about the management of those diseases in this session, because we have studied about the nutrient management, water management earlier. So this is regarding the classification only. Then there are other types of plant diseases, infective diseases. That means the diseases that are transmitted from one plant to another plant. So in these diseases, the pathogens are involved. So that means an organism, a biotic factor, is responsible for the development of infective diseases. We call infective or infectious, non-infective or non-infectious diseases. If the disease is caused by an organism or biotic factor, that means an organism is growing in the host. What is the host? Host means the organism which is affected by the pathogen or organism in which the pathogen is growing. So if a plant is attacked by a pathogen and having a disease, then that particular plant is considered as the host. So on the host plants, when an organism pathogen is growing, it will develop its own cells, tissues or organs if it is a higher taxonomic level organism. But when the pathogen is a virus, it may not develop any cell or tissue, but it will be multiplying the number of viruses. So the fungi or some higher plants, if they are infecting the host, it will develop a little bit higher organized cells, tissues, or some structures of the pathogen. So when the pathogen is developing some structures on the host, we name it as signs of pathogens. So we'll see what are the signs later. So pathogens, parts or products or structures are considered as signs of the plant diseases. For an example, we saw that there were some pictures of gherkin covered with white color mycelium or some type of a white color growth on the fruit. So that is a sign of, it is a sign of the pathogen. It is a structure of the pathogen. Then when there are some spores produced by bacteria or fungi, some oozing is there. We'll see what are those things, and we will understand using those signs for identification. Then, in addition to the signs or the structures of the pathogen developing on or in the host, there are symptoms of the plant diseases. So we were observing, we were looking at the plant disease symptoms earlier. So those symptoms are reactions of the host plant to the pathogen which is causing the disease conditions. Actually, the symptoms are created by the interaction between the host and the pathogen. For an example, wilting symptom, which we have seen earlier, it is due to the interaction between the host and the pathogen. So the pathogen is blocking the xylem vessels, xylem tissues of the host plant, and because of that, the water absorption and transpiration is reduced. As a reaction by the host, it is closing stomates, making leaves droopy, and because of that, we can see the wilting symptom. So the lesions, curling of the leaves or other parts of the plant, unusual root initiation, those are some symptoms of the plant diseases. When we talk about the symptoms, there are two types of symptoms of plant diseases. One type is the symptoms that are appearing or developed throughout the whole plant. For an example, the wilting symptom, which we have seen earlier in the picture, the whole plant is wilted. So, not only lower leaves, not only middle level leaves, even top leaves of the plant is wilted. Then, sometimes the whole plant is affected and the growth is retarded. The whole plant is dwarf. So, very low height, dwarf plants. So, similar types of symptoms are considered as systemic symptoms. Then another group, localized symptoms. Localized symptoms are the symptoms that are appearing only on certain limited areas of the plant, like leaf spots, cankers, We'll see what is fall smut later on. So the cankers, leaf spots, those are some specific types of symptoms caused by the interaction between host and the pathogen, but they are appearing only in certain areas of the plant or plant part or tissue. So that was it was a primitive, basic classification of plant diseases. That type of a classification will make us easy for controlling, managing the plant diseases. So the next step is to identify that what is that disease available in our crop. So for that purpose, there are many laboratory methods. Just for an example, we mention a method called Koch postulations or Koch postulates. That is an accurate method. Actually, in this case, if there is a time, maybe we can discuss. But in this case, the pathogen is grown on artificial medium, and once it is grown on an artificial medium, it will be easy for us to identify it through certain steps of some laboratory work and the final step in the field. So that method is named as Koch postulations. So, but that has to be done in a laboratory, in a premises, because the isolation or separation from the host and growing it on a medium should be done under sterilized condition, and it is something like growing a tissue cultured plant, but this is not a plant culture. We are culturing the pathogens. So for an average farmer, the laboratory identification will not be easy and possible because of the available condition, but identification based on some simple characteristics of the disease will be more useful or beneficial for a farmer. So those identifications or identification methods are used at the location, in the field, or just in the field laboratory quickly by using the signs and symptoms. Now you know what does it mean by a sign of a disease and a symptom of a disease. Can somebody repeat the meaning of a sign of a plant disease? And of course, if possible, what do you mean by a symptom of a plant disease? Can you please, which we had mentioned in the previous session before the break? What is a sign of a disease? Can somebody remember that, which we had mentioned earlier? So I think I started naming few. Tushan was not here. Krishna was not here. Kalpataru, can you remember what is a plant disease sign? Fatima, Ashifa, can you please? Tell us about disease sign. So, Vidurshini, only one part of the idea is given in your text. Can you please unmute your microphone and speak? Now, leaf spot is not a sign, but it's something different. Sign of a plant disease. You are mentioning is symptoms of the diseases. So, zero 0372 03219. So all are gone. Vidurshini, can you please?
Necrotic spotting, marginal scratch,
Now,
growth,
okay. Necrotic
growth,
spot, growth
growth abnormalities.
growth abnormalities, necrotic spots, they are correct.
Needle drop.
They are actually, now Vidurshini, they are symptoms of the plant diseases as a result of interaction between host and the pathogen. Those types of growth abnormalities, necrotic spots, leaf spots, lesions are developed. Then regarding the signs of the disease, signs of the plant diseases. Yes, anybody who can. Now, Vidurshini, anything now? What is the difference between a sign and the symptom? So, what you have mentioned were correct in the sense of symptoms. Am I correct in pronouncing your name?
Yes, sir.
Okay. So, what are the signs of a disease? We mentioned about it earlier during the, before the break. Anushka, what about you? There are two Anushkas, Anushka Kurupu and Anushka 3122. So change of plant color, it is also a symptom. But we mentioned that when there is a structure produced by the pathogen, in the plant, if we can see that structure, it may be some visual structure, not only reproductive organ, whatever the structure which we had seen earlier on the gherkin root or other plant part, those are signs, structures of the pathogen appearing on an infected plant is considered as signs of pathogens. Now, why I am repeating about that, the signs and symptoms are important for us to identify the plant diseases based on signs and symptoms. Both types together, they are useful for identification of diseases. So, we can consider the symptoms of fungal diseases for identification of the fungal disease. Now, these symptoms we had mentioned earlier. Then wilting symptom, necrosis, withering or wilting, little bit similar, leaf curl, abnormal curling, appearance of short hole, stunting growth, development of cankers, necrosis, that means death of cells or tissues, gummosis, gum exudation, atrophy, that abnormal elongation, anthracnose type of symptom. We'll see some of the symptoms again, the pictures, the blights and blast. So blight symptom, we saw that. Blotching is another type of blight, but without sometimes changing color. We'll see some of these symptoms later. Then if we name the symptoms caused by bacteria, they are similar to fungal symptoms. But there are no structures like mycelium or spores visible during the bacterial infection. So there are no signs such like on a fungal infection. Some symptoms like at the early stage of the disease development by bacteria, there can be if it is on a leaf, the leaf become dark green initially and then develop the water soaked area. There are rots like wet rot, some wetness and the rotting, foul odor or other types of unpleasant odor, angular lesions. Those are some of the symptoms caused by bacteria. Now, the bacterial infection in the this is an example of bacterial infection in onion bulb. So you can, I think, remember if you had the experience at least once in your life, how bad odor is produced by this. And the wetness is appearing on the infected tissues. And another thing is the development of some kind of watery oozing like symptoms. So almost all these plant parts are infected by a bacteria. That bacteria which I mentioned earlier, Erwinia bacteria. So it is not only damaging carrots, but also some other fruits through the damaged skin of the plant part. Then the symptoms caused by plant viruses. Depending on the host plant, the same virus can develop different types of symptoms. So the different ages of the host, different weather conditions, environmental conditions, nutritional status of the plant or nutritional status of the medium, soil. So those things are changing the symptoms caused by viruses. If we see some of the most commonly observed viral symptoms, mosaic symptom. So mosaic symptom, this one, I mentioned it earlier, the dark green and light green patches are alternatively developed. It is a mixture of green and yellow patches. So these types of pathogens who are causing mosaic diseases, they are causing different symptoms on different plant groups like monocotyledonous or dicotyledonous plants. And these pathogens can be transmitted to other crops by aphids and other sucking plant pests. So the viral symptom, mosaic virus symptom, I think this is more clear. In addition to mosaic symptom, sometimes with the mosaic symptom, we can see the abnormal growth of the stem and other parts. So this is one of the symptom which is very common in tobacco, tomato, potato, cucumber, gherkin, the virus diseases, specifically mosaic viral disease. Then there is another type of viral symptom we call grassy shoot. That means just like a grass, a plant is changed into the form of a grass. Due to the infection by viruses. See these rice plants, the normal plants are like this, normal height. Dwarf and stunted growth is appearing on the virus plants. Virus affected or viral infected plants are having the different shape, stunted, dwarf type of plants commonly available in rice and signature. Rice and sugarcane. Sugarcane is having many grassy shoot causing pathogens, and those pathogens are transmitted by sucking pests like aphids, thrips can transmit the grassy shoot viral disease. Yellowing, the plant is turned to yellow completely. Leaf hopper is disseminating that vector to other plants. The disappearance of chlorophyll along the vein is considered as vein clearing. That means the disappearance or degrading the chlorophyll along the vein. Death of cells, death of tissues named as necrosis. In the worst condition of the infection, worst for us or the plant, there can be some brown spot or dead plant cell areas are developed. Those places are named as necrosis or brown spots. Brown, dead spot on plants. Then some examples, the common example, ring spot viral disease in papaya. So it is causing the symptoms on the immature leaves, immature fruits. Once the infection occurred, the virus will be remaining in the infected papaya fruits, even though they are not causing any damage or infection to the consumer. Those fruits affected by viral infection is loosening the consumer preference for these fruits. Ring spot on geranium plant caused by a virus. Then pineapple wilt caused by a plant virus. Phytoplasma, like organisms causing diseases. Earlier they were named as mycoplasma-like organisms. I mean, not by our session. In other literature, it may be, it can be observed. Phytoplasma are having the common symptoms of little leaves. Little leaves disease means the leaves are developed in an unusually smaller manner. So I think you have seen the little leaf disease when the plant is affected by ring spot. It can be combining with, or effect can be combined with other pathogens effect. So yellowing symptom and some mosaic symptom can be confused with viral infections as well. Then phyllodes means abnormal growth type. The apical shoots and flower buds are converted to leafy structures. So the whole flowers and other parts of the plant are converted into leaf structures. Some common crop plants that are affected by this phyllodes disease are maize, sesame, sun hemp, like this. In the plant, there are some damages on the certain floral parts, but later phyllodes like this can be appearing on plants. So when you have a phyllodes caused by a virus, if the plant is not bearing and you can't control it by using any chemical, some prevention can be done by using like controlling vectors, keeping the clean conditions. So we'll see what are the control measures. I think we had no discussions about the symptoms caused by nematodes earlier. So it is grown on roots and above ground on aerial parts. Roots are affected by nematodes having hypertrophy. Root knots are developed, root galls are developed. Maybe the root rods are developed because of the puncture made by nematode stylet, and through that puncture some other bacterial infection can be entering into the plant tissues. So inside the cell, maybe we can see the particles are just living. As it is well developed in the evolution, I mean the nematodes or the pathogens, most of them, once they enter, they do not kill the host instantly. They keep the host live until they complete their own life cycle. That is why we can observe many of the pest remains, residues on the field or in our, premises if we handle post-harvest products. So these nodules are not the nodules caused by symptoms, but it is similar but not the same as produced by rhizobium. Now, our next task is to identify the diseases based on the symptoms in aerial parts like malformation, blistering, twisted appearance or distortion of leaves and stems. So, while looking at the symptoms caused by fungi, we need to follow several basic and essential steps. We have to take the proper sample, observing it carefully. Initial observations in the field, that is the best thing to do, because once you detach the plant part, it may be dehydrated because of the detachment. And then how it is developing naturally will be the best thing to understand. Then the spreading pattern of the disease is considered when we do field observation. And if it is a fungal disease, so we know that it can be spreaded throughout the wind, so it is spreading very rapidly. When you identify or when you are going through the identification process, you need to collect as much as possible informations, considering the crop variety, considering the crop history, what were the pesticides used by brinjal, So, using symptoms, there can be quick tests carried out. One is observing through the hand lenses. These are common with the smaller insects. Use of moist chamber. Virus particles will grow on the normal plant parts. Then moisture condition is growing. Bacteria and while fungi both are having the favorable conditions when the moisture is higher. So keeping the specimen in moist chamber will be improving the growth rate of the pathogen, and we can quickly identify what is the pathogen. Development of ooze, rubbing with water. A drop of water is released on the surface where the fungal or bacterial organism is growing. Then we can observe the fungi or bacteria within a day or two because of the available higher moisture and other conditions available. Other conditions means temperature, then sunlight or solar radiation, like that. Each and every fungus is having its own characteristics of the fungal fruiting bodies, structures or signs of the fungus. Use of the shape for identification, shape of the fungus or spores produced by the fungus under the laboratory conditions. We can transfer the pathogen into the host. So, when we isolate the pathogen, when we take or remove a part of the pathogen into a culture medium and growing it and observing it under microscope will show us some types of pathogen spores like this. So Penicillium is having characteristic type of pathogen. Spores are useful for identification. Shape of the spore is important. Then some fungi, they produce fruiting bodies. So each and every this type of structure, when it is adhered on the skin of the stem of the plant, which is affected by this, it is just like smaller chocolate balls. So we mentioned about Penicillium, Sclerotium, and Aspergillus. I think you heard about Aspergillus fungus. Even recently, there was a reference on Aspergillus fungus in mass media. What is the important characteristic, which may be a negative character, but we should know about it. So Aspergillus is produced in certain laboratories do some research work, and they are producing some Aspergillus identification kits or tests because of the necessity of identifying growth of Aspergillus. What is the reason behind that? Aspergillus is the organism that is developing and releasing aflatoxins onto the host medium or media. So the aflatoxins, you heard about it. It is grown naturally it is grown on many plant parts. Specifically, it prefers the plant organs parts with some higher oil or fat content. Then when it is produced, the spores of the fungus is produced. With that, they produce aflatoxins. So aflatoxins are serious toxin type. Recently, there was a consignment imported from a South Asian country, imported consignment of chili, broken chili pieces. So we call that, I think, a red chili imported, and it was with aflatoxins on the dried chili from some other country. And the reason behind that development is to kill the host tissues later and spread the fungus. Right, I think so maybe we can see some type of different spores. So just to have a look only, because then you know that there is a wide variation of spore shapes of fungi. These are all plant pathogenic fungi. Characteristic symptoms of these fungi, see that fungal spores and the fruiting body like this. Now we'll see this type. Can you remember the sclerotium I have mentioned? So these types of fruiting bodies will be making us easy for identification in in our conditions like that. Fusarium spores having a slightly sickle shape. So those symptoms can be compared with photos, literature. And for an example, in order to test some of the pathogens in our laboratories, sometimes some research institutions or maybe our students, farmers are sending photographs. Then based on the photograph, we can search for some literature information on that pathogen on our books, internet publications, preserved samples. And of course, all these things are important, but the experience of the grower, experience of the farmer is important for identification. So these are some symptoms, and based on the symptoms. Now this is blister blight disease on tea leaves. So I have mentioned one. What about these symptoms? Any name known to you? These two symptoms, at least, caused by a fungus named as Colletotrichum. Anybody knows about this? Leaf diseases on rubber. I think oidium leaf disease. These two common symptoms of beans and right. So as you were silent, I thought of skipping. Anyway, this is anthracnose disease on mango, a common fruit and vegetable disease. There is anthracnose spots on beans. Now these fallen infected plant parts like fruits, maybe leaves, they are fallen on the ground with the fungal mycelium or fungal structures. Now, this is a good example to show you that the earlier mentioned structures of the plant pathogen are like this. So this white color, powdery-like appearance on the plants, this sketches some few dots. Albanese pin, or we call this safety pin needle, a type of symptom is started. Later, it will be developed into large size. And the anthracnose, one of the common post-harvest diseases of many fruits caused by Colletotrichum gloeosporioides. Those are some symptoms. And yes, Vidurshini, this is similar to powdery mildew. And mildew type at these, sometimes they are in rubber, it is named as oidium leaf disease, blister blight. The blisters, they are covered with the mycelium of the fungus, and it can be mentioned as blisters, but they are signs of the plant diseases. So I would like to repeat that. If any structure is appearing, they are signs. If it is a white color appearance, it may be sign. Only color change like this, it can be symptoms. So anthracnose causing Colletotrichum diseases. So some disease symptoms like Erwinia carotivora, soft rot in carrots, soft rot in cabbage, circular leaf spots caused by circular fungus, circular type attack but caused by a fungus. Then change of the color, chlorophyll color in on the leaf blade. If it is not spreading beyond the veins, we call vein, the angular spots are produced by the pathogen or pathogenic bacteria. Leaf spots, root nodules, leaf spots, the root nodules. Right. I think regarding the symptoms, I am just showing you symptoms because we may not be able to show all these in practical sessions. Have you been to the field visit already? Can somebody give me the response? It is not a question on plant diseases. Right. So that means maybe you have not seen all these symptoms. Some of the symptoms were there. Now I thought of showing this one especially. Now, if you can remember, we were talking about disease symptom and a disease called false smut disease. Now, this is a special type of rice disease caused by a fungus. Now, see, this is a sign of that disease. This rice grain is infected. I'll start from this. The powdery-like structures of spots, spores are appearing. Now, from this grain, now see that now there are other grains, other rice grains are available on the panicle. So these spores are contaminating, disseminated into other parts of the plant. Then, now when it is appearing on the panicle, it is like this. So when these seeds, if the farmer, now this is only, not all seeds are like this. Maybe during harvesting you are missing this, you are not observing this, but you use the seeds from this field to establish the crop in next season. I think we have observed this during the practical, but I'm explaining. So when this a seed like this contaminated, or the spores are deposited on these seeds, and when they are germinating, the spore of the fungus is also germinated, entering into the new seedling during the germination, and it will be then entering into the plant, growing with the plant. Nobody can see it unless you do some kind of special test like PCR inside the plant tissues. Otherwise, the mycelium will grow inside the plant, and when the plant start production of panicle after flowering, the fungal mycelium is entering into the grain which is there, empty grain. So the field food will be used by that fungus, and it will be producing spore produced grain inside the original grain. So it is like this appearing. It is named as false smut disease. Then I use the damping of symptom drawn by an artist like this. So the stem, basal part of the stem is infected with the symptom. So the fallen leaves, drooped plant. This plant has to be removed, otherwise no point of maintaining it. It will be dying soon. Right. I think the pathogens of this damping off, they are Phytophthora. Phytophthora is the late blight causing fungus. Sclerotium, which is producing sclerotia, these types of structures in artificially grown culture. See, this is artificial culture on potato dextrose agar medium. So Sclerotium is producing sclerotia, and we can identify it by looking at this or using this sclerotia. So Pythium So different types of growth of mycelium. And there is a picture of some growth of Phytophthora inside the leaf, and the sporulation structures are appearing through the stomate of the plant. These are signs of the diseases, signs of the structures of the pathogen. Right, anyway, now you know what is a sign. Cotyl-like structure is grown. So sometimes these symptoms are similar to the symptoms caused by nutrient deficiencies. So sometimes they are similar to the damages caused by herbicides. Necrotic areas can be caused by sunburn also. Yellowing can be caused by the aging of the plant leaves, nitrogen deficiency, shade cover, and the dwarf appearance can be a character of a clone. So you know the dwarf types of plants like dwarf ambarella, dwarf plants of different species. And also it can be some secondary infections of an insect pest damage. So because of that, sometimes your experience and some expert opinion should be taken to consider whether it is a plant disease or deficiency symptom of a plant, right? So with that point, now we can see the different symptoms. Now here, these patches that change of color, anthocyanin pigment is developed. So it can be a symptom caused by a pathogen or nutrient deficiency. So for better identification, better to collect samples, preserve them in a herbarium like these two research people are collecting. And once you preserve the specimens, you can take them and show it to your maybe any extension officer in your area. Now the preservation is done now earlier. So this is how the wooden panels are there. Then on the wooden panel, a cardboard or some kind of a tough cellulose-based material is there. Then to absorb moisture, newspaper layer is there. In between that, some plants with the symptoms are placed. So that this is how this specimen collection is prepared. Then you can take this for identification of pathogen or even for identification of plants. Sometimes we collect healthy plants using these types of plant press and send it to or take it to the second opinion by an expert in an institution like maybe extension officer, research laboratory of the Department of Agriculture, like that. Right. So now we have mentioned about the classification and identification about the plant diseases. So classification and identification is important for controlling plant diseases, management of the plant diseases. So that is our next topic, which I will start again after a break, because we will have a extended session, and at the end of the next session we will have the late lunch or lunch break, ending the session, third part. So that will be starting from, yes, this time it will be, now it is 12:20, we will start at 12:30 after 10 minutes. So the next session on the management of plant diseases. So be connected. Now the original number is reduced to 75. So we'll see. We'll continue after 10 minutes.
Recording stopped.
Next topic, so we were talking about the plant diseases, their identification, because we want to manage the plant diseases or control the plant diseases. So that is our ultimate goal, ultimate objective of studying about plant diseases. So that is the next topic we are going to discuss today.
Recording in progress.
Plant disease management is started from the identification. So that initial part we have already discussed during our previous session before the break. So correct identification is very important for efficient management. That is why we were talking about the symptoms and signs of the plant diseases, how those symptoms and signs are useful for management of the diseases. And there is another thing we need to have in our mind. Once you identify the disease, whether it is a fungus, bacteria, or virus, you need to have some idea about that organism, about the life cycle of the fungus, life cycle of bacteria, or life cycle of virus. Because based on that information also, we need to take some actions. So without knowing the life cycle biology of the pathogen, it is difficult to control the disease, or your effort of controlling disease will be in vain, or you are wasting time and energy and other resources, because incorrect identification is creating problems. Then even if you have correctly identified that it is a fungus, you need to know how a fungus is growing and reproducing itself. So that should be known to us for disease management, and that basic information you need to collect. So that now when you studied about the environment, about the soil, you may have discussed about fungi, bacteria, and viruses. So different groups of pathogens are having different types of cycles, life cycles, right? So assuming that we know about their life cycles, then we can think about how to manage the plant diseases. So plant disease management is based on few principles, and under each and every principle, there are certain methods applied. So if we go through the principles and the methods or strategies applied under each and every principle, that would be, I think, better for easy understanding. So the first principle of disease management is exclusion. Exclusion means keep something away. So if I am excluding something out of my mind, maybe I would like to exclude the problems from my mind. I think you understand. So now in your houses, maybe if you don't want the disturbance from something or someone, you would like to keep that outside your house. So you want to have exclusion of that problem. Similarly, we need to exclude the diseases from our field. For that purpose, we use some strategies, methods. Now, one category of method, quarantine. So quarantine is applied sometimes at national level. There is a National Plant Quarantine Institute, National Plant Quarantine Service, and the head office or the main institution is located at near Katunayake International Airport, and their officers are working in all ports of entries, not only at airports, at marine ports, as we don't have any other entry points in our island. So there are officers who are working. So what they do is they are having some actions to prevent the introduction of diseases or pathogens from other countries. They are checking the passengers who are coming from other countries, and not only that, that is maybe only minor thing. When there is an import of plant material, seeds, grains, whatever the maybe, all types of imported items are checked, samples are checked in quarantine center to prevent the entry of any pathogen. If any infection, if any pathogen is found, the law prohibit the release of that consignment to our country. Then it will be either destroyed or sent back to the original country. So I told you about that aflatoxin in the previous session. In last week, they have found aflatoxin in a consignment of chili. So they have returned that container. Oh, I don't know, I can't remember how many containers. They have sent back those containers to the country of origin. So aflatoxin, if it is there, that means the Aspergillus fungus is also there. So similarly, if in the products or in plant material, anything is there, any pathogen is there, it is not allowed. That is the law. Then when you are exporting also, it is same. Your exported material plant material should be checked and certified by the plant quarantine service, saying that it is free of certain organisms. So that is about the national level quarantine. Then in your field of crop production, in your farm, in your plantation, in your foliage plant nursery, in your home garden, there also you can have quarantine measures. If one unit of your cultivation is infected by a disease, you can prevent the movement of people from that area to other areas. Then you can check the plant material you are purchasing, plant material you are bringing into your field, your farm to establish your crops. If anything found, you will not allow it to be used in your farm. If you are having some borders with other farms, or if you have different sections of your farm, then in between each and every section, or in between your border with other neighboring farm, you can have some quarantine rules and regulations to prevent entry of pathogen into your field. So that is plant quarantine. Then the second method of exclusion is phytosanitation. Sanitation means keeping something clean. So phytosanitation means you are keeping your plant production site clean. That means you are starting from the clean plant material. You are using pathogen or disease-free planting material for the establishment of your crop. Then how to identify that there are diseases or pests in your plant material? Of course, you need to thoroughly observe the plant material. And the other thing is, when you are collecting, when you are purchasing, when you are bringing plant material, you should collect it from the pathogen and disease-free plants or crop production areas. Then another aspect of phytosanitation is that you are keeping your field without any infected material like rotten fruits, infected plant parts. If there are any residues of crops or whatever the residues, waste material, if it is containing any pest and disease, you should collect and destroy them. Because in the residues, the life stages of fungi or bacteria can survive. So, a clean field phytosanitation is very important for controlling plant diseases. Phytosanitation. Another aspect of sanitation is that removal of weed plants. Why should we remove weed plants? How do you think? So just for a change and keep you a little bit more active, can you think the reasons to say that phytosanitation is enhanced when we remove weed plants from our field? We usually say that the removal of weed plants will be a phytosanitary measure that reduce the plant diseases or controlling plant diseases. Okay, so quickly, please let us know why weed management is important for controlling plant diseases. How can we say that weed control is a method of exclusion of pathogens or diseases. Yes, volunteers, please. I can name Achinta. Okay, I can read the number 3206. Three two zero four 3122 3080 I think we have mentioned about the weed management as a method of pest control also. So, yes, I thought somebody wanted to say. Anushka, so yes, I would like to repeat the some names. How weed management can help to manage the plant diseases.
Excuse me, sir.
Yes, please.
Sir, some weed plants are expanding some plant diseases. When we minimize the spread of weed plants, we can
We can reduce the plant diseases.
yes, sir.
So, how the weed, you said weed plants are spreading plant diseases. Some weed plants are spreading plant diseases. How it happens? Can you please little bit explain it? You are correct. How weed plants are helping to spread the plant diseases?
Sometimes weeds plants are active for some insects. You can say weed plants සමහරක් වෙලාවට අර මේ සත් يعني වගාවේ පලිබෝධකයන්ට ජීවත් වෙන වාසස්ථාන වගේ සපයනවානේ.
Okay. And then, so some weed plants are hosting some insects, and those insects are sometimes transmitting, disseminating plant pathogens like virus, phytoplasma, molecules. So they are vectors of pathogens. So from plant to plant, they are disseminating, transmitting pathogens. So if we remove those weed plants that are helping insects to survive, then it will reduce the spread of the disease. The other thing is, some weed plants are alternate hosts of the plant pathogens. When the crop plant is not available in the field, the weed plants are acting as host of the pathogen. So maybe when we are harvesting, remove the plants and then remove our crop plants, then the disease causing organism, pathogen, is surviving in weed plants. Then when we establish the crop again, the pathogen will come into the crop from that weed plant. So weed control, it is considered under sanitary measures or phytosanitation. So by doing that, we can reduce we can keep the diseases away, because we are keeping away the survival place of some pathogens by controlling weeds, right? So then some cultural practices, that means normal crop establishment and maintenance practices, are crop establishment and maintenance practices, normal practices. We consider them as cultural practices. So because of that, we consider some of those practices as crop protection or disease management methods that can be applied. So we can see some details of those cultural practices, how they are helpful. Can you give some idea, any idea about cultural practices? Now, from the starting point of the cultivation, we have many cultural practices, like we do land preparation, we do seed bed preparation, we do seed sowing. Then after sowing, we apply maybe fertilizer later. Maybe before seed sowing also we apply fertilizer. After establishment, we do many practices like irrigation, fertilizer application, pruning, training. Out of those methods, what are the methods that will reduce the pest? Suppose the land preparation, how do you think? Right, okay, so you are just silent, and it is wasting time for continuation of the session. So we will just go through the cultural methods, cultural practices that are useful for management of the plant diseases. So when we start cultivation, actually we start with land preparation. We will talk about land preparation as well. Now we select certain methods of certain types of plant material. So we use different plant materials, seeds, seedlings, vegetative parts of the plant. Then when we select the plant material, one important thing is that if we can select some plant material varieties that are resistant for pest. What kind of pest? In this case, resistant for plant pathogens that are causing plant diseases. So we can use resistant varieties, planting material of resistant varieties to control diseases. For an example, if in your area you have the ring spot viral disease of papaya, maybe you can use resistant papaya varieties like now I am naming a local variety, Ratna variety. So in the dry zone, Ratna variety is showing resistant against the ring spot virus disease. So if you select that variety, then you are out of fear. You will not get any ring spot viral disease in your papaya field. Then similarly, just I'll just go to some other slides to give you some explanation. So when we are selecting varieties, for an example, when we grow rice in rice fields, we can select different rice varieties. So these two varieties are having some type of resistance for certain diseases. So this one is having some tolerance to rice blast disease, and the LD401 is also having resistivity or tolerance to bacterial blight of rice. Likewise, you can select the resistant varieties. Then I mentioned about the resistant type of papaya to ring spot viral disease, and you can select a variety, Ratna variety, or any kind of a disease. If there is a resistant type, why not use that? Then you are sure that there will be no such disease, and you can take actions to control other problems rather than worrying about particular disease. So resistant and tolerant varieties are important. When you say resistant variety, that means that there will be no development of a disease. When you say tolerant variety, that variety can give us a reasonable yield even if the crop is infected by particular disease. Then we have mentioned another category of cultural practices, crop rotation. Usually it is considered as a weed management method, because when we are changing the crop, when we are rotating the crop, we are changing the environment. I think you can remember the session you had with Amanda Madam about the crop production systems and cultural practices. So crop rotation was mentioned during that. So anyway, when we change the crop or rotation, we are creating different environment with the change of the crop. That means the organism surviving in one crop will not be surviving in another different crop. For an example, there is a bacterial pathogen called Pseudomonas solanacearum or Pseudomonas ralstonia solanacearum. That bacteria is causing bacterial wilt in solanaceous crops. So that bacteria can infect only solanaceous plants. Tomato, chili, capsicum. So once the plants are infected, the pathogen will be in the plant. And then when we do harvesting, we know that not all plant parts are removed by us. Especially the root parts are remaining in the soil. So the remaining root parts of an infected plant, infected with the bacterial wilt causing pathogen, that plant parts will be in the soil, and in the soil with those parts, decaying parts, Pseudomonas solanacearum bacteria will survive for a longer duration. So if we cultivate another solanaceous crop in the next season, that bacteria will come back to those plants again. But if we rotate the crop and avoid the continuation of that particular variety, I mean the particular type of plant, solanaceous crop, instead of solanaceous plants, we can cultivate maybe a plant like soya bean, mung bean, that is not affected by that damping of, bacterial wilt causing pathogen. So when we change the crop like that, the pathogen survival rate will be reduced, and because of that, because of that, the next season crop will be protected from the pathogen, and the pathogen population will be regulated by crop rotation. So not only pathogens, so crop rotation is frequently done to manage the weeds, to manage the insects or other pests, as well as to manage the plant diseases. So just an example of rotation. See this field, different blocks are with different pattern of land preparation, and each and every plot will contain different crops. So we are rotating crops. Then if we practice the same cultural practices again and again, the certain wheat plants will survive better under those practices, and certain pathogens will be surviving under those practices. But if we change the crop, now for an example, here it is a different crop than what is grown here. So if I remove this crop, make it to grow here, and I put this crop in this place, what will happen? The pathogen surviving in this plot may not attack this crop, so there will be reduced incidence of the disease. And this crop is gone here, So it will not be affected by any pathogen here because it is a new field. So likewise, we do crop rotation for disease management also. There are many other benefits of crop rotation about which we are not talking much now because this is not the discussion on crop management or crop rotation, but we are talking about the use of crop rotation for management of plant diseases. So eventually the yield will be increased because of the change of the crop or rotation of the crop. So there are some more pictures like now the fields are divided into the sections. Instead of growing same crop again and again on the same field, at least we can change the varieties, or else better to change the crop in each and every field so we can rotate. Then there is another cultural practice that is called crop residue destruction and tillage. Now, first of all, what are the crop residues? Residues means like this material. So it is it looks like a maize field after harvest. So these residues may contain some growth stages of the pathogens that were damaging our crops. So now suppose that this was maize crop, and then maybe during the cultivation season, maize plants were infected by a fungal leaf disease and a fungal disease that is damaging stem and root parts. So during the harvest, we remove the pods, cobs, and the other remaining parts. Some of the parts will be fallen on the ground. Maybe some of the parts will be removed by the combined harvester. But anyway, some plant parts are fallen on the ground, and now those plant parts may contain the pathogens that were attacking the crop. So if we reduce these parts by incorporating these residues into the field, if we reduce the residues available by different methods, by incorporating into the soil, or maybe by collection and preservation, maybe using the residues for compost production, then by that way we do reduction of the pathogens in the field. And the tillage practices like this that are destroying residues or incorporating them into the soil will be reducing the diseases. Then we can do planting at the correct time. Now, see that this person is planting potato seed tubers, and tubers are already sprouted, so quickly it will be rooting. So then potato diseases some of the diseases are coming from the soil during the crop establishment. Then if we know the optimum condition for the crop plant growth during the season, and if the certain conditions of the environment or in the field are going to be unfavorable for the pathogen, and favorable for the crop, then we can select such a time period to establish the crop or to do the planting. And at the same time, when you are harvesting also, it is not just removing plants with the tubers. Better to think at what time, during what type of days, the pathogen that is causing late blight disease or Phytophthora infestans fungus, if we can select a time at which Phytophthora infestans is not much available in the field, then that would be the best time for planting and also for harvesting, because even the harvested tubers can be contaminated, inoculated by the pathogens. Then the then
Recording stopped. Recording in progress.
right. So this is a normal operation, pruning and thinning. But these operations are important for disease management also. So when there are plant parts infected, damaged by the diseases or pathogens, we can remove them, collect them, and correctly dispose them. So in a way that the pathogens are destroyed. Otherwise, these infected plant parts will be a source for infection of other plant parts. And the removed parts, if they are infected, can be correctly destroyed without
Recording stopped. Recording in progress.
Right. So something is wrong with the connection. I am at the faculty. I am having the session from our laboratory in Makandura Agriculture Faculty. Something wrong with the network. I don't know. I am disconnected. I think better to go through this quickly. Otherwise, maybe the weather is disturbing somewhere. So anyway, in addition to pruning, the removal of unwanted plants from the growing or crop production site will be important because the large number of plants will be quickly transmitting the diseases if one of these plants is infected, then you know it is quickly it will be spreaded into other plants as well. On the other hand, if the plant density is higher, excessively higher, then the humidity among the canopy will be increased, and the higher humidity will be, of course, favorable for many plant pathogens. So, not only in the plant nurseries like this, even in the field, when you establish the crop with the recommended spacing, I think I saw that one of you have mentioned about it earlier as a method of disease management. So correct spacing, maintaining the optimum level of spacing in a way that there will be no excessive humidity, no excessive increase of temperature or humidity, then it will be a benefit for the disease management as well. Right. Then another cultural practice we have listed there was the fertilizer application. Now, I think in this case, as you have already gone through fertilizer application in many modules, maybe you can help me to explain how fertilizer application can be a method of exclusion of plant diseases or a method of controlling plant diseases. Quickly. Now, fertilizer application is a normal practice. How it can improve the crop, how it can control the diseases. I hope that when you studied about fertilizer application, you were given the information that it should be balanced fertilizer application. If fertilizers are applied at lower rate, then the plant vigor will be reduced. The plant growth will be retarded, vigor will be reduced, immunity will be reduced, and because of that, lower vigor, lower immunity, the diseases or pathogens can easily attack to our crop plant. So that is one way of explanation how fertilizer application can be a method of disease management. On the other hand, if the fertilizers are applied at higher rate, then the higher doses of fertilizer will be creating toxicity to the crop, and the toxicity will be reducing immunity, and because of that, the plant can be easily attacked by the pathogens. Then another thing is higher fertilizer doses of especially the nitrogen at higher level will be creating succulence of the plant, and the succulent plants are more attractive, more susceptible for the pest attacks. So there can be more benefits of fertilizer application on time, regularly, considering the requirement. So when you apply fertilizer, you need to understand what is the crop requirement. Maybe in your field, there is no necessity of fertilizer application because your soil is fertile. But if the soil is poor, you may have to apply higher doses like that. Then similarly, the water management, another method of disease management, some diseases are easily spreaded and increased by overhead irrigation like this. So this will be increasing the humidity in the field. This method will be increasing the germination of fungal spores. Higher humidity is created, and because of that, the pest problems, specifically the plant pathogens or disease problems, will be increased. Then overhead irrigation like this using sprinklers will be not beneficial for certain plants. In that case, we have to apply irrigation through surface irrigation or submerged irrigation. Likewise, water management will be very helpful for plant disease management. Then we mentioned about crop sanitation. If there are weeds around the cropping field, and if these weeds are hosting the pathogens of certain diseases, then these weeds will be acting as alternate host of the pathogens of this crop, cabbage. Then the residues of some crops or these grasses may be harboring, keeping some pathogens, and because of that, the removal and keeping the field in a clean condition will be beneficial for the protection of our crop from plant diseases. Another method of another cultural practice, we use trap crops, and the trap crop will be cultivated maybe like this at the border of the planting plot, or it can be grown in a separate plot. Then you can activate, attract the pathogens or other pests to this trap crop. Initially, when they are coming into the trap crop, we will collect the trap crop first with the pathogen or the pest. So our main crop will be surviving because of the removal of trap crop. And during the favorable time for the pathogen, trap crop is available, and it will not come into the field to collect anything or damage anything from in our field. So trap crop, sometimes trap crop is valuable, giving us some biomass for production of some fertilizers or biological inputs like that. Then those were the methods of exclusion, and there are other principles of disease management. Another one principle is consideration of methods of avoidance. That means we are just avoiding pathogens by establishing the crop at a time when pathogen is inactive or the pathogen density or lower level is lower. Another important point is that production of the crop in areas where the pathogen is absent or the environment is unfavorable for the pathogen. When we produce planting material, seeds, or maybe vegetative propagule, it is better to produce them away from commercial production sites, because in commercial production sites, the number of plants, plant population is very high. And because of that higher population, the frequent infections by pathogens is a common thing. But when we grow plants with higher distance between the plants, maybe specifically to produce planting material, we can have some special places now. If you can remember, or if you are aware about the seed production sites of various crops, even in Sri Lanka, now in under the Coconut Research Institute, they are maintaining breeding sites in a plantation in the area of Chilau called Ratmalagara Estate, I think I am correctly naming it. And then Sri Lanka Cashew Corporation is producing and improving varieties in a seed garden established at Eluwankulama. So likewise, the commercial production sites are kept away, or planting material production sites are kept away from commercial sites. So those are some methods of avoidance of plant diseases. Then sometimes we do eradication of the plant diseases. The meaning of eradication means 100% removal of the pathogen or the disease. But in crop production, in agriculture, when we do eradication, maybe we are unable to achieve eradication 100%, or it may be very expensive, not necessary also. Under eradication, we consider the reducing, removing, and eliminating the pathogen in the source. So when we do eradication, we are trying, now for an example, if a new disease has been introduced into Sri Lanka, maybe we somehow through the air currents or somehow it has been introduced, then we are trying to remove that, eliminate that new disease. Usually when a new disease or pathogen is arrived, we are thinking about the eradication, because this is a environmental costly and financial costly operation. So especially when the pathogen is coming from a different country, when it is coming from maybe some neighboring country where the disease is there, so we have to check, and sometimes with the aim of eradication, we destroy the infected plant material, right? So for eradication, also we use crop rotation. For an example, if we take the same example of bacterial wilt causing bacteria, if I am growing crops of Solanaceae family, maybe after 5 years of time on my field, before that I am continuing with the plants that are not affected by Pseudomonas solanacearum. So as I am growing non-host plants of that bacteria, it will not survive. Maybe after 3 or 4 years time, I can leisurely, or maybe I can be sure that there will be no attack or damage by Pseudomonas solanacearum if I am doing crop rotation. Then source of infection means the places or materials where the pathogen is surviving. So I can treat that source. For an example, the residues of the previous crop can be applied with an insecticide or fungicide, or maybe the treatment is better to be done by exposing to direct sunlight. Maybe if it is possible to have some kind of heating so that the pathogens are destroyed, we can achieve the eradication. Then another action is that we can apply some eradicants, that means chemicals, fungicides or other chemicals that are destroying the pathogen. So we can apply them into the infected plant parts. And of course, for controlling the diseases, there is a possibility of using their natural enemies or antagonistic organisms. So by using the natural enemies of the pathogen, we can do biological control of plant pathogens. So there are many natural enemies of plant pathogens that can be used to control some pest. So for an example, if you can remember, we saw the names of some fungi that are causing damping off, like Fusarium, Pythium. So those pathogenic fungi are attacked by another fungus called Trichoderma. So Trichoderma can be used to eradicate another pathogenic fungi. But keep in your mind that when you are using a biological control agent as a method of eradication, there can be a possibility of surviving some amount of pathogen also, right? That is true. But we are reducing the population of pathogen drastically, and it will be highly beneficial for the crop. Because of that, we comparatively name the method of management as method of eradication. Then there are methods of protection. What is protection? Eliminating or reducing the effectiveness of the pathogen at the site of infection. That means when it is when the plant is infected, we are applying some chemicals or some other treatment. It can be solar radiation, maybe applying other aviation treatment to avoid the penetration of the pathogen into cropping system. So we are making a barrier between the plant and the pathogen by applying some method in between them, some material in between them. How we are going to do that? By applying chemicals, we can control, or we can do the protection of crop by spraying or dusting any chemical. Maybe it may not be the best option, but we can do it. Then treatment of the seeds to control the seed infections. You can have some specific treatments, maybe gamma treatment or some other energy treatments can be applied to protect from the pathogens. Application of fungicides, obviously it is clear. We can develop the resistance of our crop by applying balanced fertilizer. Reducing the crop stress will be essential to develop the resistance. So by developing the resistance, and if we can reduce the effectiveness of the pathogen, we can establish or we can reduce the establishment of the pathogen in our cropping system. So up to now, we were talking about the exclusion methods, avoidance method, protection methods, and still if we could not manage the disease or protect the crop, we can apply therapeutic methods or therapy. What is that? Curing or minimizing the losses caused by the infection using some kind of medicine. In our words, medicine, but correct term is therapeutants. Using therapeutants means the chemicals that are altering or changing the severity of the pathogen, and it is done by changing the environment in a way that it reduces the response of the plant to the disease. That means plant is given favorable conditions for its growth. So conditions are favorable for the plant, maybe not favorable for the pathogen. So actually this concept that making the environment, reducing the pathogen, it is based on a concept of the plant disease given in this diagram. Now, this is a classic example of the explanation of a plant disease. A disease is caused by three parties. It should be on a host plant, caused by a pathogen, and the pathogen and host will be living in the same environment. So they are meeting, pathogen and host will be meeting in the environment. The interaction between host plant and the pathogen will be continued if the environment is favorable. If these two are having interaction and the interaction is in favor of pathogen, there will be a disease developed. So how a disease will be developed? If the environment is favorable for the pathogen, then only the disease will be developed in the host also. So when you want to control a plant disease, what you can do, what you have to do, you can manipulate these three components in a way that disease is reduced. How to do that? How to manipulate? Just now we mentioned that we can have some actions to keep the host more vigor, strength, so host will not be easily attacked by a pathogen. Then if we can make the environment non-favorable for the pathogen, it should be done very carefully because the environment may be favorable for the host but not favorable for the pathogen. And when we do some unfavorable condition for the host, then if we apply some methods to make the environment unfavorable for the host, automatically the pathogen will attack the host. But a good crop manager, a good farmer, will maintain the environment, maybe modified environment in a way that it will be having the benefits for the host, but minimum benefits or risk for the pest. So it is not something instantly you can do. You have to plan the environment management in your field at the start of the cultivation. So this is at an illustration. The disease triangle is an illustration which gives us all ideas, all principles and ideas for disease management in the plants. So, in all types of plant pathology or plant disease literature, you will see this disease triangle diagram because it explains everything which we have discussed up to now. Disease is something, it is a result of interaction between these three parties. So, by manipulating these three parties in the agroecosystem, we can suppress the disease or control the disease. So that is the maybe conclusion of our session. Right. So thank you for your attention given. But before the winding up of the session, I would like to spend time to clarification of anything if you want. If you want to ask anything, you can ask. You can unmute the microphone and ask any question. If something was not clear, any question? Right, it looks like you don't have any question. So then we will wind up the session. Thank you very much for listening, those who were listening to me. I can remember some of you were away from the computer or your device. You are just connected only, no responses. So anyway, maybe some percentage of your group were paying attention and listening to me. So I'm thankful to those who paid attention and those who are willing to study. You can make any clarification later on as well, right? Okay, we will wind up the session. Hope you will get some idea from this session, or you have already received and using. So any question you want to ask? Yes. I saw a raised hand. Right, okay. No question. Maybe that was a mistake. Accidentally you have raised your hand.